Okay, technical problem solved. Or maybe not. You good? Okay. Our next speaker is Dean Wilcox from Dartmouth College, and this confused me, so I hope you'll explain that. I think Dean was here with us 2 years ago. I think that was his first involvement in the workshop program, and he is an expert in a technique, isothermal titration calorimetry. That's a really exciting technique. It's very interesting in terms of the theory, and it's very general in terms of what the binding interactions that can be interrogated. So I think it's really he's been an exciting addition to the program. We're happy to have him back.
Thank you, Marty. Appreciate that. I have to start with a confession. So, Serena, it's an apology to you. The lights went up because I leaned on the switch back there. So I owe you 3 minutes. It's a pleasure to be here, but it's always so hard to follow behind Serena. I mean, what I'm going to talk about is so prosaic compared to the things that she does. And, you know, it's amazing the things that she and her group are able to do. And so to bring us down to kind of more introductory general chemistry concepts, we're going to be talking about calorimetry. Probably many of you did a calorimetric measurement in your introductory chemistry class. But as Marty mentioned, this is a technique that actually shows a lot of promise for our field. And so what I'd like to do is start off by, first of all, pointing out what's going on here. My badge says I'm from Dartmouth University, and it turns out I do work at a university, but it calls itself a college, so we have to correct that here. Actually, I just dropped the name altogether. I just say I'm from Dartmouth. Okay. So 2 years ago, I gave a talk. It was 90 minutes, so I had a lot of time, and so we went into a lot of details about calorimetric methods in biomolecular science, ITC. We talked about several examples of cases that are of interest to this community, and we even touched a bit on differential scanning calorimetry. But this time, because I only have 45 minutes, and because that talk was posted, we're going to be a little bit more selective. What I'd like to do is focus only on ITC. This is the technique that the students are learning in the tutorial session. I'd like to review some of the issues about the method, just remind everyone what you can do with the method, some of the issues, red flags, sources of error, that type of thing. I want to focus a little bit on, in terms of the bioinorganic applications, this idea about the metal buffer interaction. This turns out to be a big deal in this technique. And finally, we have a more limited set of examples to talk about that will show you how you can use this method to learn some molecular insight. So, and I also want to encourage everyone to stop me with questions. Okay, this should be an interactive sort of thing. If there's something that's not clear, let's see if we can't clarify it at the time. Okay, so most of the applications of this technique are looking at binding. Okay, so for us, it's oftentimes a metal binding to a protein. In the case of Squire's research, it's SAM binding to a protein. Okay, and turns out that, you know, this is the main use of this method. There are some applications, but this is the type that we will tend to focus on. We're going to be measuring the equilibrium constant that's associated with this binding phenomenon. It gets us the free energy. We're directly measuring the heat flow, so that's the enthalpy. And so that's the strength of the technique. So you should really rely upon that data. Okay? And of course, then from that, you can extrapolate out the entropy that's associated with these binding phenomena. All right, here's the technique. Here's the method. It's a little box. Here it is over here in our custom-designed glove box, which immediately raises a point that it's important if you have air-sensitive samples to do these anaerobically, because the experiment is done in exposure to the atmosphere, at 1 atmosphere. Okay? But basically, the reaction cell and the reference cell and the electronics of these devices are very sensitive. So it's constantly maintaining constant temperature. As you add in a titrant, if it's an exothermic reaction, less heat is applied, so you get the stalactites. If it's an endothermic event, more heat is required to maintain constant temperature between the two cells, so now it's a stalagmite. So that's kind of your initial thing. Of course, the machine is computer controlled. Okay. Typical data set looks maybe something like this cartoon here. From the data, we extract three main pieces of information: the stoichiometry of binding, the enthalpy, difference between these first few injections and our final injections, and from the shape of this, we can pull out the binding constant. And here we have, as with any binding phenomena, you have a range over which you can accurately measure this value. Okay? And this, in calorimetry, these isotherms are, we talk about something called the C window. Okay? So if the binding is very tight, every bit of titrant you add binds, the titration looks like a step function. And all you can determine is a lower limit on the binding constant. If it's a very weak interaction, you get this long, very sloping type of thing, and it's very hard to, there you can only determine an upper limit. So this is kind of the range over which you can determine an experimental binding constant. Okay? And of course, you can pull out all sorts of thermodynamic stuff from the analysis of this. Okay, so in terms of using the technique, you can think about it as kind of three different levels: basic, intermediate, and advanced. You know, I think with X-ray absorption, everything is at least intermediate and maybe advanced, okay? But there is a very simple basic use of it. Is there any binding? Do you see any change as you add in your titrant, okay? What is the binding stoichiometry? You can pull that out if you see some binding. Now, the stoichiometries tend to be fairly soft, and we'll talk, numbers tend to be soft. We'll talk about that in a second. What is the binding affinity? Okay, again, it's limited by this C window, as any binding method would be. Okay? Intermediate information, okay, determining the enthalpy. That's what the technique is designed to do, but there's a caveat. You're measuring all the heat of everything that happens in that cell when you add the aliquot. So you got to be very careful about interpreting that, and oftentimes, certainly in our field, there's going to be some analysis of the data afterwards to pull out the enthalpy of the molecular phenomena that you're trying to measure. Is there coupled deprotonation or protonation that accompanies binding? Turns out something we've kind of found to be particularly insightful is this competition between metal ions and protons. Okay, you can imagine this is kind of pretty important to biology. And the technique of isothermal titration calorimetry can be used to quantify proton-coupled deprotonation and protonation. Okay? Eventually, of course, you can pull out the entropy of this information. And, you know, I still don't know what entropy is, but, you know, we get a number for it. Okay? But the caveat here is that you have to make sure that the free energy and the enthalpy are both measured under the same conditions. You know, if your binding constant is done under different conditions, you can't compare it to an enthalpy under different conditions. So that's just one caveat. More advanced things are determining the change in heat capacity of binding, effects of other perturbants. This gets into really kind of the detail of the biophysics of things and less concern to us. And eventually, maybe 2 years, we'll come back and talk about using ITC to measure kinetics. Okay, but that's kind of not pertinent today. Okay, what to do with the data? Okay, so this is some of the data like the students are collecting this week in the tutorials. Okay, certainly you want to subtract off any heat of dilution. Man, you measure heat when you titrate water into water. So there's always going to be some heat associated with just adding a titrant into a solution. Okay. Now, you can talk about doing a controlled titration where you don't have, say, your protein in the cell, but it's much better if you, in fact, take these last inflections out here. No further binding, and that, in fact, is your true heat of dilution for that experiment. And you subtract that off all of the data. You integrate the peaks, integrate the area, integrate the heat, and therefore then you end up with the data here, which is normalized on kilocalories per mole of injectant. Now I step back and say, let's think about it, okay? What about the stoichiometry? Is it what I expected? What about the injection peaks? Do they look, am I capturing all the heat of each injection, okay? Is the data within the C window? Is there enough heat? If there isn't, you maybe need to decrease the concentration. If there's plenty of heat, lower the concentration. Get more data out of that precious sample that you've prepared. And clearly, this is kind of what all of us have to do, right? Initial data, better data, optimal data, reproducible data, and of course, we publish the very best data, right? All right, quantitative fitting of the data. Now, here's where a lot of, in fact, in the tutorial, the students are actually going to be working with fitting some data using some of these simple binding models. Okay? The ones that are commonly available are just a single site binding model, two independent sites binding model, a sequential binding model. This tells, all this comes out of, different equilibrium relationships. And one that we'll talk about a bit later is a competition binding model. Okay? When you have two things competing for one thing, you have to, it's a little bit of a different mathematical analysis. And if you know the binding constant enthalpy with one thing, oftentimes you can use that to pull out that for the competing entity B. Okay? And then, since what you measure is experimental binding constant and experimental enthalpy, these are condition-dependent values. Then you have to think about, okay, what do I need to factor out so that I can report condition-independent values? All right. Technical issues, red flags, okay? Noisy data, okay? Oftentimes you might find there's a precipitate or an air bubble that can lead to kind of noisiness, turbidity, if you will. Irreproducible results. Cleanliness, very close to godliness in this field, okay? Clean the cell, make sure it's nice and clean, make sure your samples are clean, there aren't any interfering things. Again, air bubbles, sometimes the irreducible results are the chemistry that you're studying. So sometimes it can be a clue that you find, okay? Low net heat, okay? Remember, this is the enthalpy of everything going on, and there are cases where all the enthalpies that are happening cancel each other out, and you have very little heat that's measurable. Now, luckily, mostly these binding enthalpies of phenomena that take place in aqueous solution tend to be temperature dependent, okay? Delta Cp is not zero, okay? So oftentimes if you shift to another temperature, that will then, in fact, affect these enthalpies and can lead to an increase or even a decrease of a signal. Check the conditions. You go to a different buffer. Question here.
Is it fairly easy to change the temperature within the box?
Yes.
You want to run it at body temperature?
Yes, exactly. So good point. Most of the data is collected at 25 degrees. That's kind of standard conditions. But 37, fine. Up 40. And oftentimes when you're looking at a temperature dependence, oftentimes you'll run data from 10 up to 40 at different values. Okay? But remember, it's isothermal, so all your data is collected at one temperature. Okay? Let's see. Slow kinetics. Well, remember, it's stirring. That's a little paddle stirs things, so you get rapid mixing. Speed up the paddle. Make it spin faster, or go to a higher temperature. Okay. Sources of error. I mentioned the stoichiometry is a softer number. That's because it depends upon two concentrations and different volumes, the volume in the cell and the volume that you're adding with each aliquot. So there's a lot of places that error can come in there. 0.8 is usually 1. Okay, 1.2 is usually 1. Okay, but 0.5 is probably 0.5, and you might want to think about that. So that's a number that tends to be softer. The binding enthalpy, remember, you're measuring the net heat flow. Okay, so a lot of things may contribute to that, and we'll talk about that a bit more. The binding constant, remember, you're measuring the overall equilibrium between the titrant and what's in the cell, and it's very useful to make sure you compare and confirm this with another method. Turns out ITC got a bad rap early on when it was being used because people weren't being careful, and so the binding constants that were being reported from ITC didn't match those from equilibrium dialysis, and people said, that technique sucks. And luckily, if you're careful, the method will, in fact, do that. So that's a very important confirmation. Okay. Okay, now metals. All right. So metals offer their own challenges. Okay. Metals are Lewis acids. Buffers are Lewis bases. There is no innocent buffer. They're always going to have some type of interaction between the metal and the buffer. And that interaction has to be broken apart before the metal can bind to whatever it's binding to. We know all about this stuff here. Oxidation of metals, redox chemistry, hydrolysis chemistry in solution, precipitation dissolution. These are things you want to avoid because heat's associated with any one of them. Okay? And these are, if you want to just measure binding, you have to be careful to make sure you eliminate these. Certainly under anaerobic conditions, we'll avoid this. Precipitation dissolution, if you can keep things soluble, that's good. Hydrolysis is a little bit insidious and tricky to see, but sometimes you can identify that. Finally, this idea about metals competing with protons. Once the proton is displaced, it's going to bind to the buffer, and there's a heat that's associated with what you measure. Okay, now talking about, question? Okay. So focusing on these last 2 points, okay? So this idea about pH and this coupled equilibria, these are very common, okay? If you're looking at protein binding to DNA, there's probably less proton flow that's associated with that, but we do have to worry about that, and you need to account for it. This buffer, okay? So choice of the buffer can be really important, not only for maintaining pH, but the buffer-metal interaction can suppress side reactions, like this hydrolysis. If you have a well-defined metal complex you're delivering, it can suppress side reactions and then give you more accurate data. Can provide a competing ligand for the metal. We'll see how we can use that. But you need to know the metal-buffer interactions. Finally, this idea about interaction with protons. Okay.
If you don't have a competing metal ion, does that help to make things a little nicer?
If you don't have a competing...
A metal ion that's part of what's going on, does that make things a little nicer? I mean, a little less complicated?
If the metal's not
Yeah. So, well, if there's interaction with the metal ion, so if there is no metal ion that's involved,
right.
I would think that would make it nicer.
Right. Oh, yeah. I mean, if this was the Biophysical Society, we wouldn't be talking about this stuff. Okay, this is really metallo-specific ideas here. Okay. Okay. Now, the analysis goes back again to general chemistry, Hess's law. Everyone remember Hess's law? Summation of enthalpies. So basically, we're measuring, the overall enthalpy of the reaction, which may involve multiple equilibria. So we have to account for all these different enthalpies of the metal buffer interaction, protonation of the buffer, to finally get what we're interested in. Okay. So this is the type of a post hoc analysis that we need to do. All right. So here's the experiment that we're doing in the tutorial. We're doing very simple. We're taking copper ion and binding it to albumin. Okay, albumin, of course, very abundant, almost dirt cheap. Okay, turns out it binds copper and nickel at this N-terminal site, these first 3 amino acids, which are not seen in the crystal structure because they're randomly disordered. But in fact, this is the coordination that we find. Okay, notice we're binding to the imidazole. It's deprotonating 2 amides. Pretty strong Lewis acid to do that. Nickel and copper will do it. Okay, binding to the N-terminal amine. How many protons do we expect the copper or nickel will displace from albumin when it binds? 3? Close to 3? 2 from the amides and 1 from the N-terminal amine. Okay? So that's going to be a factor here. Okay? Now, turns out this has been measured for a long time by many different methods, and let's look at the stability constant, the equilibrium constant associated with that. At pH 7.4, the log K by all these different methods is somewhere in the 10 to the 12th, 10 to the 13th. What is that? Femtomolar? I mean, that's really high affinity. Okay, there's a few outliers there. How are we going to fit that if your C window kind of stops at 8, 10 to the 8th? Okay, well, here's the idea. We're going to use a buffer that binds to the metal fairly tightly to compete with the protein. Okay, and so that's going to allow us to have a binding constant that is measurable, that falls within the C window. So that's going to be the key tool here. In fact, here's just what the data looks like. Okay, this is copper binding to albumin. Okay, K value is like 10 to the 12th. Okay, but it's in the C window because it's competing with tris buffer, which has a fairly high stability constant with copper. So this is kind of like the general ideas. Now, this buffer interaction, you may say, well, how am I going to find that information? Okay. Well, just a tip as to how a lot of this isn't available in the databases. Okay. So what we've been doing in my group and other groups have been actually taking measurements of metal ions binding to very well-known ligands. Here's TEPA and DTPA. These are kind of simple amine ligands for which the copper ligand stability and enthalpies are well known. So now we're going to do a titration. We're going to titrate the ligand into the copper, or it could do it vice versa. They bind in 1-to-1 stoichiometry, but in the presence of the buffer. So the thermodynamics that we get out of the fit here is, in fact, if we compare that to what we get to binding with this, the difference is, in fact, the copper interaction with the buffer. Okay? So this is a way that we can actually start to develop this information for this post hoc analysis. Okay? And here's for a series of buffers. Here's the data that we find in TEPA and DTPA. The stability constant. Notice these are log betas. We don't really care what the stoichiometry is. We have a large excess of the buffer, but we find beta value, stability constants that are fairly similar and enthalpies that end up being fairly similar in many cases. So basically, this is the data that we can use for analyzing our data when we work with metals in this buffer to subtract out the buffer interaction with the metal. And just something that we recently published from my students in Methods in Enzymology, which basically has a big table of this type of information. So it's a good resource for pulling out that information. Okay, lunch is coming up. Let's talk about a few examples here. Okay, now, one of the first metalloproteins that Serena introduced to you yesterday morning was one of the blue copper site, blue copper proteins. And we've done some measurements of copper binding to azurin. Azurin is one of the best studied blue copper proteins. Remember, this protein does one electron redox chemistry, cycling between copper 2 and copper 1. And we've been interested in what are the thermodynamics of the copper binding in the two different oxidation states. So we'll talk a bit about that. We've been collaborating with Rachel Austin on looking at metals binding to metallothionein 3. Show you some of that data, again, because it's interesting and complex, but we've been able to sort out what's going on by applying these kind of techniques that we've been talking about. And finally, okay, are there any journal editors in the audience? No, there aren't any. Okay. I'm going to do something that's quasi-ethical, quasi-unethical here, okay? So I was asked to review a paper by a journal editor, okay? You know, it's sworn to secrecy, okay? But this paper had some ITC data in it, and I thought it would be very instructive if all of us kind of looked at it together and thought about critically what is this, you know, what can you say about it and what, in fact, maybe have the authors overinterpreted, okay? Now, all identifying information has been removed. This is anonymous, but I think it might be useful for all of us to do that. And we won't tell the editor, will we? Okay, azurin, okay? Here's the structure of azurin, a beta barrel structure. Here's the copper binding site. Here's the coordination. You remember there's 3 strong ligands, 2 histidines, and a cysteine, kind of a trigonal coordination. In azurin, it's a distorted trigonal bipyramid because there's this axial methionine, long bond, and this axial carbonyl of this glycine residue. So that's not, you know, very good coordination site for copper 2, or probably even for copper 1 very well. But this is the site that we're going to be binding our metal to. Okay, now I want to draw your attention to 2 other amino acids, these 2 histidines. They're kind of remote from the site. They're not even in the second sphere. They're just kind of nearby. Okay, keep them in mind. Now, here, in fact, overlays the active site of azurin with copper 2, copper 1, and the apo. And you can see, and this is the point that Serena made, is that this site is tuned up for very little reorganization between the 2 oxidation states, so very fast electron transfer. And that's shown here. I mean, the ligands are all essentially in the same position in copper 2 and copper 1. A little bit of difference in apo, but not that much. So the question here is, the metal ion binding in 2 different oxidation states to the same coordination site imposed by the protein, what is the thermodynamics? How is it similar? How is it different? This is the question we want to ask. Okay, let's see. Here we go. Okay. We weren't the first people to ask that. Okay? So back, 12, 13 years ago, Jeremy Berg published copper 2 binding to azurin. Now, it's very appropriate that I present this data, because isn't this Berg Auditorium? It's not Jeremy Berg, no. Okay. But Jeremy Berg, I mean, amazingly smart guy, head of NIGM for many years, recently appointed editor of Science magazine. I mean, a real pioneer. And so I really want to give him credit for saying, you know, here's something that ITC can do. It can tell us information about metals binding to proteins that are very well studied, but we learned something new about the thermodynamics. Okay, here's the data they published. Notice that the injection peaks are biphasic. They took a lot of time in analyzing those injection peaks to pull out the kinetics of that. Now, we're not going to be concerned about the kinetics. We're concerned about the thermodynamics. So here's the data, okay? Now, what do you think about the C window? Inside or outside the C window? That's kind of a step function, right? Pretty tight binding, all right? So, how did they analyze the data? So it turns out they were collecting this data in this very unusual buffer. Anyone ever heard of choline chloride buffer? It's a quaternary amine buffer, and with this positive charge, the assumption was that it didn't interact with the metal very much. Okay? So that was the buffer they basically had to use to collect this data. Okay? Now it turns out we've actually reproduced this data by doing the exact experiment they did. You're titrating the enzyme, apoprotein, into the copper. Okay, so you got 1 millimolar azurin in the syringe, and you're titrating that in, and you got the choline buffer, and you can see we've got reproduced their data. Very steep inflection. We can see the biphasic injection peaks, but this is pretty unusual conditions. We don't usually titrate the protein into the metal, and this is a weird buffer, as we found in studying it. So here's what they reported, an enthalpy of minus 10.2. Now notice that's right off the data. That's the delta H ITC, right? And they reported a K value of 10 to the 13th, which is pretty big, okay? So how did they do that? Well, again, I mentioned they were very creative in how they solved this problem. They said, let's look at the entropy. Let's take some very standard information about the entropy process, metal dehydration, translation enthalpy, conformational entropy, which is pretty close to 0, calculate a delta S, use this delta H, and from that pull out a binding constant. Okay, so hopefully you can see there's a number of problems with this analysis now because they're using the experimental enthalpy, and this is kind of a backdoor way to get at things. So we figured there must be a more straightforward way to quantify this. In fact, by looking carefully, we've actually found conditions under which we can do a kind of a more standard metal binding experiment, copper into albumin in different buffers, and notice that that's a weaker binding, but that's well within the C window. Okay? And when we think about this, what's going on here, let's all together think about this. We've got copper interacting with the buffer, azurin, probably some protons are being displaced from that thiol, okay, at least. Copper will bind to the protein, and then we've got the protonation of the buffer. So there's all these things going on that are leading to the heat that we measure. Right? So this is the sum of it all, okay? And the key thing here is M. How many protons are being displaced from azurin when copper-2 binds? Because that adds this heat contribution, which can be sizable to what you're measuring. Okay? Here's what we're interested in, the enthalpy of the deprotonation of the protein and the metal binding. And so you've got to correct the experimental value with the metal buffer interaction and the buffer protonation times this number of protons. How are you going to do that? Well, it turns out it's a very simple relationship called falls out of this. Okay? If you perform the experiment in multiple buffers, then you can plot the experimental enthalpy plus the metal buffer interaction times the buffer protonation enthalpy, and the slope is M. Pretty easy, okay? So, well, it wasn't easy. A lot of work went into collecting this data. Multiple data sets. Okay, but here it is. Here's a plot of the buffer protonation enthalpy versus the experimental enthalpy, and you can see from the slope we get about 1.5 protons are displaced from azurin when the copper-2 binds. Okay? How does that match with what we might expect? Okay? Well, it turns out azurin is so well studied, we know the pKa's of the important residues in the apo form, in the copper-2 form, and the copper-1 form. So we can actually make some predictions. So in the apo form, the cystine's protonated. One of the histidines at the metal site has 0.8 protons. The other has a very low pKa. And these two histidines here that I mentioned, we're going to have to keep track of them. Copper binds, it turns blue. Clearly we're going to lose these two protons. This pKa is modified slightly Okay, and so if we do a proton inventory, we predict from pKa's that, in fact, about 1.7 protons should be displaced from the protein. And we measure 1.5. So pretty close, but match between experiment and what you predict from other experimental data. Okay, so now taking that, we can do our thermodynamic analysis, the Hess's law analysis, pull out the enthalpies. We measure the binding constant. We know the stability constant with the buffer. We can subtract off that. And so we can end up with data for copper-2 binding to azurine. Here it is in 4 different buffers. And here's the log K value. Pretty tight binding, I would say. Okay. Now, let's look at the thermodynamics. Enthalpy, +2. Wait, that's endothermic. What's going on here? Well, look at the entropy, -23. Copper-2 binding to azurine is entropically driven. Enthalpically, it's not a very happy copper ion binding to that site. Okay? Remember also, there's an enthalpy that goes into deprotonating that active site. You have to enthalpy of deprotonating the thiol and that imidazole. So we can summarize. We can say the coordination site enthalpically is not very favorable for copper-2, but entropically binding is very favorable. We're going to desolvate the copper ion. We're displacing protons, and there's little rearrangement, so no penalty for that. So interesting. Not maybe what we might have expected with copper-2 binding to azurine. Okay, copper-1 binding. Now, copper-1, of course, is trickier. All right, you got to worry about oxidation. You do it in an anaerobic environment. You have to worry about disproportionation that occurs with copper-1, disproportionating to copper-2 and copper-0. Fairly favorable. So we're going to use the copper-stabilizing ligands. Okay, that will help to suppress that side chemistry that what we want to have to worry about. We've found that this ligand, hexamethyl triane, in fact, is a very good copper-1 favoring ligand. These methyl groups enforce more of a tetrahedral coordination, and you can measure the stability constant, which is fairly high, and the enthalpy. Notice here that when copper-1 binds to this, there's still some protons bound. When copper-2 binds to hexamethyl triane, all the protons are displaced. It's a much stronger Lewis acid. Remember, copper-1 is a weaker Lewis acid. So the protonation state is different than what you might originally suspect and what we originally thought. Okay, copper-1 binding to azurine. Okay, so here's the experiment. We're titrating copper-1 stabilized by hexamethyl triane into the protein. Here's the binding curve. It's tight, but that's well within the C window, so we can pull out do that for our analysis. Again, same set of contributions. We now have to worry also about this ligand being protonated as it's being released from the copper. But we do the same type of analysis here, and we come up with the same type of issue. We're going to have to quantify the number of protons that are displaced when copper-1 binds. Okay, same type of analysis here. So here's our plot with different buffers. And the slope now is 0.3. Okay, smaller slope. Less protons are displaced when copper-1 binds. What about comparing it to what we predict? Well, again, copper-1's bind, I guess azurine goes from clear to gray when copper-1 binds as opposed to blue. We're going to displace the protons from the active site. The hexamethyl triane has to pick up a few protons. Notice when copper-1 is bound, it affects these 2 pKa's noticeably. So in fact, there's some protonation of those that take place when the copper-1 binds. We do a proton inventory here. We come up with a prediction of 0.9 protons going to the buffer. We measure 0.3. So there's a bit of a disconnect here in terms of one experimental measurement from calorimetry, another predicted from experimental pKa's. Still wrestling with that question, but here's an area where different methods should be converging on the same analysis. Okay, copper-1 binding to azurine, different buffers, log K, about 2 orders of magnitude higher than copper-2. To be expected? What's the reduction potential of the blue copper proteins? They're positive, about 300 millivolts, so you'd expect it would favor copper-1 over copper-2. All right, so overall stability, let's look at the thermodynamics. The enthalpy, enthalpically very favorable binding for copper 1, still entropically favored, but not nearly so. Okay, so we can say the site is in fact enthalpically more favorable for copper 1. Copper 1 entropically, there's desolvation, but maybe less than there was with copper 2. Still have displaced protons, but remember these two histidines, they're being protonated from the buffer. So that's an entropic penalty. So all this comes into this. So basically what we found is that copper 2 and copper 1, very different binding thermodynamics when they bind to this very same site. Okay. Two more quick little stories. Okay, one has to do with metallothionein. This is a protein that probably people in our field know about. Small cysteine-rich protein, binds dye-positive metals, very thiophilic. Probably zinc. It may interact with zinc in the absence of these toxic metals. Binds the metals in these clusters. There's these two lysines that are kind of the link between these two domains that bind the 7 metal ions. Rachel Austin is very interested in one isoform of metallothionein, metallothionein 3, which is found in neuronal tissue, the brain. And we've been working with her. It's a little bit different than the metallothionein that's found in other tissues. A couple of key things. One is in this beta domain, there's these two prolines, which would affect the metal binding properties. And also there's this insert in this region, making the alpha domain a little bit larger. Okay? So the question is, this protein has a very high affinity for lead, may play a role in lead neurotoxicity, or maybe protecting from lead neurotoxicity. So the question is, how tightly does this isoform bind lead versus zinc, which is kind of maybe we think about the native metal, if there really is one for metallothionein. Okay, so what we did with Rachel Austin and her students from Bates was we looked at some measurements. First of all, we took lead and titrated it into zinc-bound metallothionein, and we see a displacement experiment. Okay, and this is one data set, but the average is about 7, so each lead displaces 1 zinc. We can pull out, now remember this is the displacement, the difference between the affinities for these two metals and the enthalpy associated with that. Try as we might, we couldn't get an experiment to work where we titrated the metal into apometallothionein. You think about all these cysteines, readily oxidized. The other problem was the samples were prepared in Maine and the measurements were done in New Hampshire. And, you know, different states have different rules about experiments, whether they work or not. So. Anyway, so we couldn't do the direct measurement. So what we did was we fell back on a type of measurement that had worked successfully in some other cases. And that is to start with the metal-bound form and titrate in a chelate that extracts out the metal. And now you base your analysis on microscopic reversibility. Okay? Now, here's the data. Whoa! Not so straightforward, right? Well, I see 3 inflections here, right? The EDTA is chelating out 3 different populations of these lead ions, right? Sequentially. And that's what this data is telling us. So how are we going to analyze that? Well, we work with a kind of a limited set of data of fitting protocols, but we in fact were able to use them in a unified sort of way. First of all, we did a 1 set of sites binding to this first inflection. Then we did a 2 set of sites binding to the second and third. Now these two, in fact, allowed us to get good numbers for the enthalpies. Okay? Turns out for the binding constant, it's more tricky because now we got a competition between EDTA and the metallothionein. So we have to use the competition binding model, and that only is a 1-site model. So we had to use that for this last inflection here. We get a binding constant of about 8 times 10 to the 11th. Now the difference here is about 2 orders of magnitude. So we know that this is the last lead that's taken out. That's bound most tightly. So the others, in fact, are bound less tightly by about 2 orders of magnitude. So that's how we can kind of estimate what's going on in this case. The analysis is a little, whoops, where's the analysis here? The analysis is a little bit tricky. This is the chelation. We have to worry about the EDTA. Now we've got this in terms of binding. The first ones that bind, second one, third ones that bind, and the stoichiometry associated with that. And in fact, we can pull out the thermodynamics of this in terms of the sequential chelation of the metal ions. And in fact, we end up with a thermodynamic table that says, you know, the binding here, again, is mostly enthalpically driven for both lead and zinc. Okay, lead binds more tightly, and that's the analysis from that. Okay, finally, okay, now, cone of silence is going to descend upon the room here. Okay, so this was the paper that I was asked to review. These scientists, very creative work, really amazing stuff. By using phage display, they expressed and selected for peptides that bind lanthanides. You could see there are some implications there. Turns out they bind the lanthanides as hydroxo species and form insoluble aggregates. And the potential applications, of course, would be to mine lanthanides from seawater, remove it from wastewater, if you could selectively precipitate it, basically. The aggregates and their formation have been characterized by an alphabet soup of methods. How many of these can you recognize? Okay, FTIR, some of these are elemental analysis, microscopy, XAF, so that's one we know, NMR, Kara's going to tell us about NMR, and ITC. So here's their model, okay? The peptide interacts with the lanthanide, +3 ion. Deprotonation, they showed that the interaction leads to a lowering of the pH. Aggregation, and then finally precipitation. Notice a single arrow here, okay? Here's their ITC data, okay? These peptides, they're known as LAMP. 2 of these were selected with diprosium, and 1 was selected by neodymium. And here's the ITC data. What does that tell us? Stalagmites, endothermic binding, right? Okay. Look at there's different enthalpies of dilution. Look at the stoichiometries. Okay, so they're fairly high. This is a peptide that is known to bind disoprisium, but not to precipitate. So this is kind of a control, kind of a simple binding experiment. Okay, so this was their data. Okay, and now, what did they say about that? Well, to analyze the data, they assumed that the binding was 1 metal per 1 peptide. Turns out there are other data from these other methods that shown there's actually 2 bind per peptide. Well, okay. What's the difference between 1 and 2 between friends? What about K value? Okay, here's the equilibrium constants that they report for the binding. But if this involves a precipitation that's irreversible, what does K really mean? Okay. Remember, it was enthalpic binding? Okay, so here's the enthalpy that they report from their data, and then here's the entropy that they get from that. Now, if my K value is suspect, and I measure my experimental enthalpy, I don't know that I put much faith in the entropy associated with that. So the lesson here really is, I'll just summarize that in my very last slide here, is that for this particular case, the correlation between ITC data and molecular processes can be challenging when it's a complex system. So don't try to overinterpret the data. Here are just some of the points that I made from the study with azurin and the study with metallothionein. And with that, I'm going to end and thank some of the people that did the work. Here's the triumvirate, Nick, Ann, and Colette, that started us off with really careful analysis. 2 years ago, Molly and George were here to lead the tutorials. This time, it's Michael Stevenson and Michael Kukin. Stand up, guys. There they are. We're leading the tutorials. And with that, I'll thank you for your attention.
Questions for Dean? I have one. So back to the copper azurin story.
Yep.
In principle, you can create a thermodynamic box where you could check the internal consistency of your 2 binding constants with the difference between the reduction potential of, well, I guess it would be free copper. I don't know how you would do that, and the reduction potential within azurin. Can you do that?
Yep, you're right, Marty. In fact, I'm writing up a paper on that right now.
Oh, okay.
I'll include you as a consultant on that. No, that's exactly true, because if you get the thermodynamics of binding, you know the copper reduction potential. So you can pull out then the enthalpy and entropy of reduction of azurin. Really neat how you can do that. Good point.
Hold on. Get the mic.
So I know that some iron proteins, so the apoprotein has no metals bound that iron can bound, and on top of that, like a water ligand will coordinate. So, can you account for that? Do you need to account for that, or how do you do it?
We're not so good at quantifying water ligands. That's a little bit hard one. Clearly, if it deprotonates and there's a proton associated with it that's lost, then we can quantify that. In fact, we've taken that to look at enzymes that involve hydrolysis. Oftentimes, enzymes that involve metal hydrolysis involve a metal hydroxide species as the active form. We can actually pick out when you get the active form, but that's a tricky problem.
Okay.
Other questions?
I'll be around all week if you've got more
I
questions.
guess people are hungry, so go forth and eat and enjoy
Everyone's
the
hungry.
practicals this afternoon. Thank you again.